Assalamualaikum warahmatullahi ta'ala wabarakatuh, everyone. Today we're going to continue with the genetics 2. So we have been discussing about the genetics 1 in the previous class, and I hope you stay well during this pandemic situation. And let us continue with our lecture for genetics 2. As for the previous class, we have gone through about the Mendelian genetics, and today we want to focus more on the human counterparts. So in the first part of the genetics, we have learned about the construction of the Punnett square and also some of the important issues during the genotyping and also for the phenotyping of the garden peas. And you can see there are some dominant and also recessive traits that we have discussed. So as for today, we will take a human sample to illustrate this, and we will try to define the inheritance and explain the inheritance of dominance, plus the recessive and some of the complex inheritance, as well as the sex-linked traits. So for that one, we might go through with the meiosis and also mitosis processes. So what is inheritance? It's the passage of hereditary traits from one generation to the next. It's the process by which you acquire your characteristics from your parents, and you may transmit some of the traits to your children. Okay, so it's inherited from generation to generation. Whatever phenotypes that you are gaining from your ancestors, it can be further expressed towards the last generations. Okay, so you have like a similar facial features, you have similar color of the eyes, you have similar shape of the bone. So these are all the inherited features. So the branch of biology that deals with inheritance is called the genetics. So genetics is very wide field. So we have discussed about the plant genetics. We have discussed we also can learn about the bacterial genetics. We also have the virus genetics. Okay, so we have the human genetics. There are lots of branches of genetics, and most of them are dealing with the inheritance, how you can determine whether some of the genetic material can be transmitted from one generation to the offspring. So the area of healthcare that offer advice on genetic problems, or if you have issues or genetic modifications, so you need to do the genetic testing, and part of this you need to undergo the genetic counseling based on the discussion with the geneticists and also the physicians. So inheritance, the nuclei of all human cells except gametes contain 23 pairs of chromosomes. They are diploid number, so 23 pairs of chromosome. You got 23 pairs from your mom, 23 pairs from your dad. So the total chromosome for normal human is 46. Okay, we have 46 chromosomes, or 23 pairs of chromosome. So one chromosome in each pair came from your mom and the other half from your father. In each of these two homologous contain genes that control the same characteristic. For example, the chromosome 1, okay, the first pair, which is chromosome 1, you have chromosome 1 from your dad and chromosome 1 from your mom, and both of these chromosome 1 inside the cell functions to express a certain similar characteristic. They share the similar genes. Okay, they also have the similar length. Okay, in terms of the length of the chromosome, they share the same size. All right, but as you go through to the bottom one towards the end of the chromosome, for example chromosome 20, you can have like a shorter length of the chromosome. Okay, this is what we're going to discuss in the next few slides. And if one chromosome of the pair contains a gene for body hair, for example, its homologue will contain a gene for body pair in the same positions. Okay, so you always have a pair, so you one from mom and one from your dad. Okay, the chromosome must be in pairs. It's compulsory to express a certain gene. Then this is called as the homolog chromosome, one from your mom and one from your dad. And at each genomic loci, okay, loci or location, you can find that these gene, okay, certain area or certain location of the gene functions to control certain phenotypes. In this case, the body hair, okay, the eyes color, okay, the skin pigmentation. So they share the similar gene. It's just that which gene is dominant. All right, is it the one from your dad, or is it the genotype from your mom? So it always come to the decision where you cannot decide. Okay, sometimes you will know your sister can look like your dad, because some of her chromosome may have inherited, okay, may have a dominant trait inherited from the father side. Okay, maybe you, if you are a boy or you are a guy, a male, then you may have some features from your mom. Okay, more with the with a fair skin. Okay, with a whiter skin color. So some of these are possible. Okay, regardless the gender. So this is what we want to learn today. So if you look at the chromosome structure, it looks like here, like a X, okay, the letter X. But if you go through, if you can see from the gross appearance here, there's a one structure in the middle. This is what we call as the centromere. Okay, the centromere is the place or the point where these short arm and the long arm intersect. Okay, so in the chromosome we have like the P arm, we call it P arm, which is the short arm, and also they have the long arm, we call it the Q arm. Okay, so P arm and also the Q arm. So each of these site or location, they have a certain gene. Okay, for example, gene that controls the hair color. So you have here from here to here, and maybe at the bottom arm here, okay, the Q arm, maybe they determine the what we call the size of the bone, for example. Okay, maybe the gene that functions to control the development. So if you go through with the microscope, you can see there's lots of supercoils, and these are all the genetic materials being compacted into histone structure, where you can find the DNA double helix structure. And each of these single location can have a different function. Okay, they may encode for different type of protein, so that you can make the structure of your body. All right, so it's very important from chromosome 1 to chromosome number 23 to be properly regulated, so you will not have any genetic disease. If you are exposed to any carcinogenic agent, for example, it may create some mutation to the DNA sequences, and it can lead to the cancer and some other inherited diseases. So alternative form of a gene that code for the same trait and are the same at the same location on homologous chromosome are called the alleles. So the alleles are the same trait, okay, the same features, the same characteristic that you've got from your mom or dad. So these are called the alleles. Okay, genetically we call it allele. On the right hand side here, you can see the arrangement. Okay, we call it as the karyotype data from your single cell structure. Okay, this with a very small single cell, you can have 23 pair of chromosome. Okay, each single cell in your body, for example, if you have hair cells, you have the bone cells, you have your liver cells, inside the DNA they contain all of these 23 pairs that makes you as a human being. So we should be surprised with that. Okay, and among of these 23 pairs of chromosome, 22 pairs, the first 22 pairs are called the autosomes. So what are the autosomes? Autosomes are the somatic chromosome. They are the chromosome that makes up your body cells, your body protein. Okay, for example, they can make your kidney cells, they can make your eyes, they can make your skin cells, okay, the cells for the body, okay, they can make the organs. So the other part of the chromosome, which is the last chromosome, we call it the chromosome number 23. So we call it as the sex chromosome. Okay, these are the germ cell chromosome, okay, the sex chromosome. These chromosomes are very special. Okay, it determines the gender of the person. All right. So if it's here XY, it can say, you can say that it's a a male. Okay, it's a male. And if it's double X here, if it's XX here, then it does mean it's the female. Okay, so we will go through and try to understand this mechanism. So one allele of the previously mentioned body hair gene might code for coarse hair, and another might code for fine hair. It can be like the coarse hair is the dominant trait or the dominant alleles, and whereas the fine hair is the a recessive allele. A mutation is a permanent heritable change in allele that produces a different variant of the same trait. So as mentioned earlier, so if there's any changes in the DNA, okay, could be due to the pathogens, could be due to radiations, it can be due to some chemicals, so it can make your DNA altered and also mutated, and then they can disrupt some of the proteins for the formation of the characteristic. In this example down below here, you can see like the blue one, this is the recessive for the blue eyes. Okay, it's very rare. Okay, if it's recessive, it's very rare, and the gene is not strong enough. But for the brown eyes, you can see the dominant allele. Okay, the dominant allele most of the time. Okay, you can see that the people or the person have the dominant brown eyes. Okay, this is the example of the heterozygous heterozygous dominant. All right, because heterozygous, they don't have the same dominancy. Okay, they don't have the same what we call allele or traits. In this and the next example, this is the homozygous, but this homozygous is dominant. Okay, so the person, okay, the offspring may have the brown eyes. Okay, the other type, which is the homozygous recessive, this is however is very rare, but it is possible in the population to occur. Okay, so what is genomic imprinting? Most genes give rise to the same phenotype, whether they are inherited from the mother or the father. Most of the genes, okay, most of the genes. In a very rare case, however, the phenotype is dramatically different depending on the parental origin. Okay, the variation is there, the possibilities is there. If you can see, if you have like lots of siblings, maybe you have 10 siblings, you can see the variation that arises. You can say some of your siblings, they look like your mom in certain features, and some of the sibling may look like your father in some of the characteristics. So these are the various genetic variation that you can observe in your family. um, the genomic imprinting, okay, this is very surprising phenomenon. This is first discovered in 1980s, where most genes usually, okay, in under normal condition, we inherit two working copies, okay, half from your mom and half from your dad. But with the imprinted genes, okay, for some reason we inherit only one working copy, not two working copy. Maybe the working copy is from your mom only, okay, but you lost the other copy, and maybe the working copy comes from your dad and you lost the one from your mom. All right, so depending on the gene, either the copy from your mom or the dad, okay, it's epigenetically silenced. When you say about the epigenetic, it's the environmental alteration of the genetic material. It's not the environment that you understand, but here is the environment that introduced inside the cells at the gene level, okay, at the cellular level, where there are some what we call as the group, okay, the chemical group, biochemical group, for example, methyl group. You have maybe some acetyl group. You have like a nitrogenous group that can't change the composition and also activity of the genes. All right. In this case, silencing usually happens through the addition of the methyl group during the egg or sperm formation, and some of the ethyl group also participate in this mechanism. So the epigenetic tags on imprinted genes usually stay put there. Okay, means stay there for eternally. So they will stay there and then keep on passing this information to the next generation. But for some reason, they are reset during egg and also sperm formation, regardless of whether they come from mom or dad. Certain genes always silence in the egg, and the others are always silence in their sperm. So this is an example of genetic imprinting. So if you say genomic, this is also referred to the whole genome of the human. But if you say the genetic imprinting, it refers to a particular location of that chromosome. So in human, the abnormalities most clearly associated with mutation of imprinted genes are Angelman syndrome. Okay, the topmost, Angelman syndrome, and how these can be detected. How you can know whether this is the Angelman syndrome? You can observe from the physical features. They have some mental retardation. You have ataxia, you have seizures, and you have minimal speech. Some of these are related to the cognitive function, and these results when the gene for particular abnormal trait is inherited from the mother, okay, not from the father. So these results in only one working copy. If you can see here, the P in blue is the paternal, the one chromosome coming from your dad, and the M is maternal, the red one come from your mom. So the location is in the Q arm, okay, the long arm, at location at the location 15 Q13. That's the location inside the chromosome where you can see the normal genes that control the normal cognitive function. But in the case of Angelman syndrome, you can see since it's only have one working copy of the gene of the chromosome, you can see only the parental, okay, only the dad's chromosome is working, the other copy is not working. So this is mainly happen inside the maternal, okay, maternal deletion. It occurs in 65 to 75% of the cases. So in Angelman syndrome, if I ask you in the examination, so what is the characteristic of the Angelman syndrome? Maybe you can say that it's the deletion at location 50q13 in the mother. Okay, and then I will try to change the question into in the father, for example. So it's quite easy for you to detect. So you need to know about this. How about the paternal UPD? So this is very rare occasion where you can see both copies are the copies of the parents of the dads. You lost the copy from the mom. And also some of the mutation of UBE3A gene. So it occurs in 5 to 11% of the cases. and imprinting defect occur around 3%. Similarly, on the other hand, if it's occur in the dad, okay, it's called the Prader-Willi syndrome. It has some physical features like the short stature, mental retardation, obesity, poor responsiveness to external stimuli, and sexual immaturity that results when it is inherited from the father. Okay, under normal condition you can see the same location, but if the deletion occur in the father, it can lead to the Prader-Willi syndrome, and then you can see both copy at 20 to 30%. You will have both mom, okay, maternal chromosome, and also between 2 to 5% possible for you to get the imprinting defects. So these are the example of the genetic imprinting. Dominant alleles for severe disorders. Alleles that code for normal traits, they do not always dominate over those code for abnormal ones. But dominant alleles for severe disorders usually are lethal and cause death of the embryo or fetus. So it's very important. The dominant genes or the dominant alleles are the main player in this. In most of the cases, if you have mutation in the dominant allele, you will have a very severe disorder or severe genetic diseases. So it's very important that these dominant genes are preserved. The dominant genotype is not mutated. So for example, okay, one exception here is Huntington disease. It's also a one type of genetic disease that causes the progressive breakdown or degeneration of the neurons. So what happens is that the Huntington's patient may have cognitive infunctionalities, okay, disabilities, and it can cause movement disorders. It can cause the psychiatric disorders and many others. Okay, the Huntington disease is caused by a dominant allele whose effect do not become manifest until adulthood. So you cannot know since it was born. Okay, maybe externally, okay, you can see from the physical appearance they appear normal, but when they reach the adulthood, there is the period where you can start to detect for some of these symptoms, and you can know that this is the Huntington's disease. Both homozygous dominant and heterozygous dominant people may have exhibit the disease, but the homozygous recessive are normal in this case. All right, so the HD causes progressive neurodegeneration and can also be fatal, but because the syndrome is not appear until they reach the adulthood between 30 to 40 years old, many people with this disease may already pass on the allele for the condition to their children. So it's very important for you to do the genetic testing. All right, so even though you think that you are healthy now, okay, between 20 to 40, 30 to 40, when you want to get married, you need to check for these neuro degenerative impairments. All right, because these diseases may only manifested when you are growing a bit older. All right, so you don't want your kids, your child to have the same problems. So make sure you get married to those people who do not have this Huntington disease. So based on the genetic mechanism, if you want to compare, if you have the big H here, okay, the Huntington's allele is a dominant with the capital H, and the other one you get from either your mom or dad, which the normal allele, which is the lowercase h. In other words, this is the carrier. So you want to get married with the normal people, all right, without any capital H. But if you get married with the one with the capital H, the heterozygous dominant, or could be the both capital H, which is the homozygous dominant. It can cause most of your kids or potential children to have this Huntington's disease. Let's have a look. Even though you are the carriers and you do the genetic screening, and then you see that your spouse do not have any of these Huntington's allele. So if you do the cross, you can see there is some possibilities that you can carry this Huntington's allele towards your offspring, even though you are the carrier. Alright, so it's very important. This is an example of the hereditary traits in human. You can see on the left hand side is the dominant features, and the right hand side is the recessive. You can see that the normal skin pigmentation is the dominant trait, but for albino people this is very rare and recessive, where their skin lost the pigmentation capabilities. Near or farsightedness, this is very dominant. People with the eyesight problem, but the recessive is normal visions. Okay, so you can say that in this population, more people have the eye problems as compared to the normal vision. So those people who, for example, did not wear the eyeglasses, maybe they are the recessive groups. Ptc Taster, some people have the capability to taste a special chemical compound, we call it phenyl thiocarbamide. It tastes like a very bitter. Some people don't have the capability to taste it. It comes like from the plant sources. Alright, so people might have or might not have this capability. So for in the dominant trait, they can taste the PTC most of the time. Okay, most of us may taste the PTC, but rarely that people can have the ability to do the same. Polydactyly, extra digit in the offspring is very common. Okay, very dominant. But normal digits is very recessive. Brachydactyly, dominant syndactylism, where digits, some of the digits can combine with each other, like the pointed finger can combine with the middle finger. Okay, they don't have the gaps in between, and so this is the dominant, and some of other example. Chromosome mutations and non-disjunction. Occasionally an error in cell division, we call it as non-disjunction. It's the result of abnormal number of chromosomes where they divide or where they segregate unequally. So in this situation, homologous chromosome during meiosis 1 or sister chromatids during anaphase or mitosis or meiosis 2, they fail to separate equally. All right, so you know that inside the meiosis you need to know to separate the haploid gametes in equal proportion. But in the non-disjunction, okay, in the non-disjunction, these are not the case. Okay, they will segregate unequally. A cell from which one of the more chromosome has been added or deleted is called an aneuploid. A monosomy 2n minus 1. Okay, this is the monosomy. In other words, there's only one chromosome for that pair. Okay, for example, in chromosome number 10, there's only one from your dad, or it can be only one from your mom. So this is what we call the monosomy. Okay, however, on the other hand, if there's an addition, okay, there's an addition of the chromosome number, for example, you only have one pair, or you have two only chromosome, okay, but in this case, in the trisomy, you will have another addition of the chromosome. Now it becomes 3, so it's called the trisomy chromosome. So this extra chromosome can come from your dad or from your mom. It also can come from the error during the cellular division. Most cases of Down syndromes are aneuploid, right? Aneuploid disorder in which there is a trisomy of chromosome number 21. Okay, you already came across the Down syndrome before. Maybe you know the features of the kids, or that can also can also be observed in the adulthood. There are the changes that occurs in the chromosome number 21, where you can find there are 3 chromosomes available instead of normal 2 chromosome only. So the non-disjunction usually occurs during gametogenesis, meiosis, okay? The cellular division in the germline or the sex chromosome involved are the meiosis, whereas the mitosis only occur in the somatic cells. Okay, but in this case, only 2% of the Down syndrome cases result from the non-disjunction during mitotic division in early embryonic development. All right, so one instance, okay, the first causes of Down syndrome are the non-disjunction. All right, remember that. Okay, so this is the example of the karyotypes from your single cell spread of the chromosome. Okay, you can see the autosome, which is the somatic chromosome from chromosome 1 to 21, from 1 to 22, and chromosome number 23 is the sex chromosome. You can see this could be the male person. Okay, this is a male sample because it's contain X and Y, and if it's contain both X chromosome in the chromosome number 23, it leads to the a female person. Okay, but I wish to highlight the Down syndrome cases where you have the trisomy. You can see here there are available 3 chromosomes here, and this is very unusual. All right, so this is very unusual. So it's 2n, okay, the diploid 2n plus 1, so it's called the trisomy. If it's 2n minus 1, it means it's the monosomy. So how does this happen? In the process or in the cellular division of the germ cells, you can see in the meiosis 1, under normal circumstances, they separated equally, and each gametes contain half from your mom and half from your dad. But in the non-disjunction, in the case of the possible Down syndrome, you can see the shorter chromosome and also the longer chromosome here in yellow, they combine together and they did not equally divide. So it will continue to only segregated inside the first cells, whereas the other half during the cell division, they don't have the longer chromosome, so they have lost it. So what happens next? In the gametes, you may have the trisomy. You can see here, you have one of these, and one of these, and each cells of these have the trisomy number of chromosome, and the other half just pass on what the one that they have only. Okay, so this one is the monosomy. You have 2 cells that contain the trisomy and 2 cells that possess the monosomy chromosome. In some other cases, whereas this in the anaphase 2, okay, the non-disjunction can occur in anaphase 1 here, and it also can cause during the anaphase 2. For example, if it's occur in the anaphase 2, you can see that the chromosome here, the longer chromosome in yellow circle here, they did not divide equally. All right, to become or to produce the gametes. Okay, on the other hand, they divide equally and give the haploid number of gametes, and this increasing the chance of this this single cell to become the trisomy and the other half become the monosomy. So there are two possibilities that can happen at two different stages of the meiosis to contribute to the trisomy and also monosomy appearances during the cellular division. On the other hand, okay, like in the Down syndrome, instead of the non-disjunction, which is considered as the primary Down syndrome, the Down syndrome can also caused by the translocation of the chromosome. What are the translocation? Okay, the translocation can be passed on. Okay, it can be inherited if the Down syndrome patient grow older and they want to get married, and after that they will have this probability to pass on their genetic material to the offspring, okay, by the translocation method, not by the non-disjunction. All right, the non-disjunction is not inherited. Okay, it can only occur at one time only, but in the case of translocation, it is possible. So that's why in the non-disjunction cases, we call it as the primary Down syndrome, whereas if it's translocation that occur, we call it as the familial Down syndrome that can be passed on to the next generation, and maybe they can be the carrier. Okay, and this is not the case in the primary Down syndrome. So what is translocation? Is another error in meiosis. In this case, two chromosomes that are not homologous break and interchange portion of their chromosome. All right, they may be intersects in between, and they change some of the genetic material naturally. Okay, we do not decide on this, but it happens naturally. The individual who has a translocation may be perfectly normal if no loss of genetic material took place when the rearrangement occurred. However, some of the person's gamete may not contain the correct amount and type of genetic material. Okay, sometimes, okay, when this translocation occur, okay, they may have a normal features. Okay, maybe we all already have this translocation inside our chromosome. Okay, but we are not expressing that genetic material. That's why we don't have the Down syndrome symptoms. Okay, so this is the one that is carried out. Okay, the familial Down syndrome. This very rare and occur during the translocation. About 3% of Down syndrome cases result from a translocation of part of chromosome 21 to another chromosome, usually chromosome 14 or 15, and the individual who has this translocation is normal and does not even know that he or she is a carrier. So as I said, even though you have this translocation at chromosome number 21, okay, you may not have the Down syndrome. Okay, in other words, you can say that it is very less people having this translocation. Okay, you will not become a Down syndrome, but with one possibilities is that if you get married with the other carrier that also have chromosome 21 translocation, you are increasing the chance for getting an offspring with the Down syndrome. All right, since both are the carriers now. So when such a carrier produces gametes, however, some gametes end up with the whole chromosome 21 plus another chromosome with the translocated fragment of chromosome 21. Upon fertilization, the zygote then has 3 rather than 2 copies of that part of chromosome 21. This is example of the balanced and also unbalanced translocation. You can see you have the chromosome A and chromosome B. Inside the Q arm at the bottom here, some parts they can be interchangeably changed. All right, they can share these genetic materials. Sometimes they intersect. Okay, bertindan ataupun dia duduk ke atas ni for some reason, okay, during cellular division. However, this will contribute to a normal or balanced translocation. In the case of unbalanced translocation, in this example, you have this long, very long chromosome, okay, but they are not properly in a proper or in a balanced proportion. So these chromosome of parent 2 with the balanced translocation, then this chromosome parent 1 is the normal. However, when this balance meet the normal one, it can contribute to the unbalanced translocation. So this is just example of the Down syndrome caused by the translocation, where the chromosome in the chromosome number 21, they translocated, okay, they've been translocated into the chromosome number 14, okay. So they change some of the lower arm, which is the Q arm genetic material, and then they take the chromosome 14 Q arm and put it into their chromosome. And consequently, in the offspring, you can see some of the offspring 1, 2, 3, they are not viable, maybe they died, and one of them become Down syndrome, and some of them have the balanced translocation carrier that will pass on this genetic material to the next generation and possibly cause the Down syndrome too. Okay, so this is the chromosome number 21. Okay, this is not the non-disjunction, this is the translocation. You can see or some of the chromosome in 21, okay, the trisomy chromosome translocated to the upper arm of the chromosome number 14, or it can be in the chromosome number 15 too. However, the people that have this will not have the facial features like the primary Down syndrome cases. Variations of dominant recessive inheritance. Most patterns of inheritance, they do not conform to the simple dominant recessive inheritance, in which only dominant and recessive alleles interact. Okay, there can be some variations occur. The phenotypic expression of particular genes may be influenced not only by which alleles are present, but also by other genes and by the environment. So variations, when you are dealing, when we are talking about the dominant and recessive inheritance, it may include the incomplete dominance, multiple allele inheritance, and also the complex inheritance. So these are the possible variations, okay, due to the dominant recessive inheritance. All right, it's not simply if you say that your dad have the dominant allele, it's not necessarily expressing that dominant allele all the time. Okay, it can also be some other different mixture of characteristic that you can see. All right, maybe it's not coming from your mom and dad, maybe they come from your granddad, okay, or maybe they come from your granddad. In your many siblings, maybe you can see some of your siblings have a very special traits or unique characteristic that are different from the others. So these are example of the variations in the dominant and also recessive inheritance. The first one is the incomplete dominance. It happens when neither member of a pair of allele is dominant over the others. Okay, no expressions, or that's an absence of dominant allele in this case. Okay, we call it incomplete dominance, and the heterozygote has a phenotype intermediate between the homozygous dominant and the homozygous recessive phenotype. So we call it like the intermediate in between. Half of them are dominant, half of them recessive, so they are not sure. It's 50-50. It's a mixture of both. So we call it as the incomplete dominance. For example, in the sickle cell disease, these are example of the incomplete dominance. You can see here on the right hand side, okay, example here from the sperm, and also from the ovum. Okay, you have the sperm from your dad and the ovum from your mom, and these HB, the homozygous dominant, or the hemoglobin for the absence of the sickle cell disease. Okay, you have the heterozygous. Okay, heterozygous dominant. The same goes with the ovum, heterozygous dominant. So your mom has heterozygous dominant ovum. Your dad have the heterozygous dominant sperm, and these two fertilize together. And if you tabulate the Punnett square, you can see that actually you can get half. All right, you can get half of the offspring having the half of the offspring may, because this is the sickle cell disease. So uh, although they are usually healthy, those with the heterozygous, okay, HBA and HBS, heterozygous dominant, okay, have minor problem with anemia because half of their hemoglobin is normal and half is not. Okay, so in this first box, okay, it's homozygous dominant. Obviously it's contain healthy blood. Okay, the heterozygous dominant, okay, these two, okay, they have the minor problem with the anemia, but in this case they have this sickle cell anemia. All right, so it's the incomplete dominance. Okay, you can see from the phenotype, okay, half of the offspring may have this anemia, and the, not half, okay, it's 3-1, because it's this this is dominant, and dominant, and this is the recessive. Okay, only the recessive have the sickle cell anemia. Two of them are the carriers, and one is completely normal. This is another example of the incomplete dominance in the plant. If you fertilize these true breeding red color flower and the true breeding white color flower in the first generation, you can see combination of both. And if you mating these two gametes with a similar generation of the similar features, you will have these 1-to-2-to-1 genotype. But in terms and in terms of the color, you have 3 different colors. Okay, the red, the pink, and also the white color flower. Okay, so you have now a new features. Okay, the combination of both. So in these two, we can say that they are the incomplete dominance state. All right. How about the other variations in dominant recessive inheritance? We have the multiple allele. Although a single individual inherits only 2 alleles for each gene, some genes may have more than 2 alternative forms. This is the basis for multiple alleles. So when you say about the multiple alleles, so you have more than 1 allele, it can be more than 2. Okay, for the nearest example is the blood group. For example, we have 4 different blood group, 4 different blood types, okay, A, B, and O. So they result from the inheritance of 6 combination of 3 different alleles of a single gene called the I gene. So for example, in the allele I, with the capital A here, it produces the A antigen. Hence the final outcome is the blood type A. Okay, how about with the B? So it produces the B antigen. If it's with the lowercase I, it produces either one. Okay, either A or B, not O. Okay, so we'll see this. For example, if a person inherits two I gene, okay, the capital I gene allele, one from mom, one from dad, it will give rise to many phenotypes, and the possible genotypes produce 4 blood types as follow. Okay, how many phenotypes? Okay, so sorry, how many genotypes? 6. Okay, you have 6 genotype. 1, 2, 3, 4, 5, and 6. How many phenotypes? 4. 1, 2, 3, 4. So this is the only possible genotypes and phenotype. So if you have the capital I gene that encode for the antigen A, so if both are the homozygous, or if they are heterozygous A, it can give rise to the blood group A. Okay, if it's homozygous B or heterozygous B, it can give rise to the blood group B. If it's combination of A and B, it can give to blood group AB, and if both are the lowercase I, it can give to the recessive blood O type. So when we say about the multiple allele, so this is what we mean. It can be a combination of two different allele in one trait. Okay, one I gene. Okay, one genotype. So it can be two different features, okay, or two different traits or alleles that we are discussing. Notice that both IA and IB are inherited as dominant trait, and I, the lowercase I, is inherited as the recessive trait. And because of an individual with blood A, B group has characteristic of both A and B, okay, the combination of both, we call they are said to be the codominant. Okay, codominant means that they share the dominancy by half. Okay, half is belong to the A and the other half is belong to the B dominant. All right, so they are now equally heterozygous, and depending on the parental blood types, different offspring may have blood types different from each other. Okay, due to this multiple allele, and the blood group is very special and very unique, even though your mom and dad in a different blood type. For example, your mom has A and your dad has B group type, you may have the O blood type. Okay, this is possible based on this theory. All right, so as I said earlier, you can see here, if your mom got A blood type and your dad got B blood type, the possible offspring may have AB, or an O. Right, why this happens? So look on the right hand side here. All right, so you can see the A and O here in the chromosome. Let's say you inherited, this is your father chromosome. Okay, the blood A from his mom. Okay, for example, O from his dad, and the same goes from your mom. B from his mom, her mom, and then O from her dad, and this is combined. You can see that you will have some, okay, half of this. Okay, maybe the first child have the AO, second sister you got the AB, and the other one BO, and then the last one got the OO. So the one that we want to highlight here is the codominance. It is possible, okay, for the A and this A to be expressed together, and give rise to the blood group or the antigen A and B at the same time. Okay, they have expressed two of these um, antigens in an equal proportions. Okay, this is what we call as the codominance. All right. Uh, the last one, what is the complex inheritance? Most inherited traits are not controlled by one gene, but it's very complex. Okay, it can be combination of two or more genes. Okay, multiple genes. A situation we call it as the polygenes or many genes due to the environmental factors. All right, so this is called as the complex inheritance. You can change your DNA composition due to these factors. Okay, example of complex traits are like the skin colors, hair colors, the eye colors, the height, metabolism rates, and body build. And in complex inheritance, one genotype can have many possible phenotypes. Depending on the environment, or one phenotype can include many possible genotypes. For example, even though a person inherits several genes for tallness, full height potential might not reach due to environmental factors such as disease or malnutrition during the growth years. So again, even though you have a very good genotype and you think that your mom and dad express the phenotype very clearly, but it is possible for you of not having some of your phenotype from your mom or dad. Okay, this is due to the environmental factors. If you got disease, it can change your growth. Okay, if you having or you lacking vitamin D, it can lead to some bone problems. All right, so it really depends on your lifestyle, your diets, and it's very complex. All right, so these are three examples of the variations in the dominant recessive inheritance. Okay, the possible variation, of course, it's genetics, but the genetics can be changed by all of these factors. As I mentioned earlier, for example, the skin color. Okay, these are the example of complex trait. It depends on the environmental factors such as the sun exposure, nutrition, and other genes. All right. So this is just explanation what we have discussed previously. Okay, you can have a look and understand this, and let me know if you did not understand any of this. All right. In the sex-linked traits, autosomes, sex chromosomes, and sex determination, you can see how do we be either the male or female genotype. Okay, if you view under the microscope, there are 46 human chromosomes in normal somatic cells that can be identified by the size, shape, and staining pattern to be member of 23 different pair. Okay, we call this as the karyotype. An entire set of chromosome arranged decreasing order. Okay, the first chromosome pair can be longer, and as they going through towards the end, it's become shorter, and it should be pairing of this. You need a cytogeneticist to do the rearrangement and then do the pairing for all of these chromosomes. All right, so in 22 of the pairs we call it autosome, and the last pair we call it as the sex chromosome. It can one X chromosome also present in males, but its mate is much smaller chromosome called a Y chromosome that only have 231 genes, less than 10% of the 2968 genes present on chromosome 1. Okay, you know that if the chromosome is very short, you know that the gene can be lesser. Okay, so you can say that genetic or the genes inside the chromosome 1, okay, have a higher number of gene as compared to the to the chromosome number 22. Okay, and you need to know that in our genome, in our body, there are more than 20,000 genes. All right, okay, so in the chromosome there are more than 20,000 genes that have different functions for the development, okay, for the cell cycle, cellular division, for organ development, for embryonic or fetal development, for brain functions, or many other functions. All right. And individual sex is determined by the father's chromosome. The secondary oocyte is fertilized by egg-bearing sperm. The offspring normally is female XX, here in the bottom here, okay. And if fertilization by Y-bearing sperm produce the male, all right. You cannot decide about this, okay. It occurs naturally. So if you do the Punnett square again, so you can see what would be the possible outcomes during the fertilization. You have the XY from your sperm, from your dead sperm, and you have the egg XX. If you do the Punnett square, you will have the equal proportion of male and female. So that's about the sex links, inheritance, how you become, how the cells decide whether it can be XY or XX. And the example for the sex-linked traits are the red-green color blindness and male pattern baldness, hemophilia, Duchenne muscular dystrophy. All right, so these are all the traits that can only occur if the issues or error happens inside the chromosome number 23, which is the sex chromosome. In the red-green color blindness, this is the most common type of the color blindness. The condition is characterized by deficiency in either red to green sensitive cones. So red greens are seen as the same color, either red or green, depending on which cone is present. The cone is the inside your eyes, okay. That's the receptor for your eyes to see any or detect any wavelength, okay, the color wavelength, okay. The gene for red-green color blindness is a recessive one, designated by the lowercase c. So in this image, if you have the from your dad, okay, you have the capital C, which is the normal, all right. And if it's fertilized with the homozygous heterozygous dominant, you can see if you do the Punnett square, you have one normal female, but as a carrier, one normal male, and also the color blind male. All right. So what do you see now? If you have the color blindness, maybe you will be not able to detect this left hand side fruit and also this beautiful lady here. Okay, it will appear something like this. All right, I think that's it for today. Thank you very much for your listening, and please let me know if you did not understand one of the lecture. It's raining heavily right now. I will provide the I attend for you to sign in before you attend the lecture. I think that's it. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.